and provide any updates that you'd like to provide. But in the chat, just let us know who's here, and also let us know what kind of topics you want to hear. For this meeting and for the May meeting, we've heard a lot of requests to have a variety of people present findings of work they're working on. And so that's what the next this meeting and the May meeting will be about. So I'm not going to talk long because we have a lot of great people to hear from. I do have one question to cover. If let me get to my next slide. Let's see. There we go. We've had a number, and this quick question is about our work group name. We formed ourselves as the Puget Sound Research and Monitoring, Kelp Research and Monitoring Work Group. And that followed on the Kelp plan that came out, which was focused on Puget Sound. And I've had a number of requests to change our geographic focus to the Washington State Kelp Research and Monitoring Work Group. And I haven't heard a single person say that we shouldn't do that. So I was hoping to ask this group, would anybody is there anybody who would like us to consider not changing the name to the Washington State Kelp Research and Monitoring Work Group? And I haven't heard a single request to leave it. So I just figured it'd be easier to see if there are people out there arguing to keep the smaller geographic focus. The rationale for extending it, of course, is that culturally we want to be inclusive, and also ecologically, the coast is really important, as we're going to hear today from Nick. Okay, I'm going to take that as a consensus that we should update the name. So now we're going to be the Washington State Kelp Research and Monitoring Work Group. And I'm going to pass it on to Elizabeth to start working us through the agenda. Welcome, everyone.
Thanks, Helen. Okay, so we have a few updates before we move into our presentations. So I know we are just getting started, but do we have Cynthia Harbison and Max Showalter with us today? Have they signed up? There she is. I see her. Okay.
Hi.
I'm going to hand it over to you.
Thank you. Good morning, everyone. My name is Cynthia Harbison. I'm a planner in DNR Aquatics, and I'm co-leading the development of the statewide kelp forest and eelgrass meadow health and conservation plan with Max Showalter, who I believe is not going to be on this call today. And also, at the risk of sounding like a broken record, for those of you on this call who have heard me talk about this many times in the last few weeks, we are hosting a kickoff workshop next week, which I'll put the information in the chat to help. It's our kickoff for our development of the prioritization framework to help us identify the at least 10,000 acres of priority kelp and eelgrass habitat that we'll be working on. That will be, we will be focusing our actions around over the next, I guess, 20 years. So I really hope that folks on this call can attend. And if not, we also have a survey that we'd like that folks can fill out if you're unable to attend. And we also will be having an additional workshop in March. We're actually having a Puget Sound focused workshop and a coastal focused workshop. So there will be additional opportunities to provide input as well in that series of workshops also. So thanks for giving me the floor, Elizabeth, and I'll put my email, Max and my emails in the chat as well if you want to reach out directly to us. Thank you.
Great, thanks. And for those who do have the agenda in your email, those links should be workable. So if you need to go refer back to them, they should be in there. Okay, Dana, you are up next. So we are going to hear from Dana Oster with Northwest Straits Commission on the recommendations that were developed through the Pew study linking kelp science and policy.
All right, thank you for having me. Hopefully you can you see regular mode and not presenter mode?
Yep, it looks good.
Awesome. So as Helen said, everyone's got calls to share results. So I've got a different flavor of results to share with everyone in the research and monitoring work group, which is a check back to the kelp conservation and recovery plan, which we often point to in this work group. And the words we used was that the Kelp plan provides a framework for coordinated research and management actions. And so after the publication of the Kelp plan, I went on a journey to look at that coordinated effort and the management action side and how that links together with the research and also the knowledge gaps that we know we have and identified in the Kelp plan. And recently, Tom Mumford shared with me that there's actually a name for what we were doing and are attempting to, well, we're not necessarily successfully doing, but it is our aspiration, which is translational ecology, which is the approach using ecologists and decision makers to work together to develop research that address the sociological, ecological, and political context of an environmental problem. So if you're curious, go Google translational ecology and ways that we can work together to integrate both the research community and how science can inform policy. Part of this journey is that we've talked a lot about stressors driving kelp, and there's certain things we know, but there's a lot we don't know locally. And that kind of spirals into this whole web of how do we actually, what levers do we have available to us to control those stressors and in hopes reduce what is driving declines in kelp in the Salish Sea. And so we've got a lot of these great conceptual frameworks like the one that Jordan Holler-Smith put together of drivers and those states that they affect and pressures that puts on kelp. And then we've also started discussions of, well, what are the actual tools and management entities we have in Washington state to control some of those local impacts? And so we've got this slide prepared by Don Gourley that kind of identifies those resources as 3 simplified buckets of water quality, nearshore habitat, and food web, what the impacts are and what the management tools are that currently exist. A lot of this was because of the creation of the Kelp plan when we were starting to dive into the management side. What we heard a lot is that there probably are certainly gaps in policy, but there's a lot of language that exists that is not necessarily completely successful in our ability to actually use it for kelp. And so that is where a lot of this work started to focus. I'm going to show you one more kind of squiggly chart, which is the Kelp plan, which is a great resource, is also a really great resource to dip your toes into the management context in Washington state. So I also want to direct you back to chapter 4, which has both this figure that Max Calloway put together and outlines of all the different regulatory authorities within Washington state and how they play together. And so again, if you want kind of a brain teaser of a different flavor, go back to chapter 4 of the Kelp plan. And where this took me was Pew Charitable Trust has been a great partner in funding this work of looking at the regulatory kind of implementation and authority and how it's actually working relative to kelp. And so this work included some multiple workshops and an advisory committee that is still continuing on to take on this great work. And I also held a lot of one-on-one discussions with folks in different authorities, so local jurisdictions, state agencies, federal agencies, and tribes and nonprofits and some others to understand how are the regulations actually being applied in practice and what resources are being used, and more importantly, what are the needs to support actually implementing the rules and regulations we have in place. And so from all of those conversations, the workshops and everything else in our advisory committee, we have honed in on this is really one piece. When I pointed to those 3 broad buckets of water quality, shoreline modifications, and food web, a lot of what we started to tackle was really at the site scale. And so that's individual permits, small parcels. It's not necessarily tackling the cumulative impacts, which is often discussed and I'm sure might come up later. But this is first really focusing on what are the barriers and challenges that people have just at the site scale for implementing rules and regulations we have. And the 2 main things that were identified were that there's the processes or sources used for verifying that kelp is present or absent at a site are inconsistent, vary across jurisdictions, and in some cases, there is no process in place. And additionally, this is a really big one. There is limited guidance for reviewers, so those administering rules and regulations, to 1, verify impacts to kelp from proposed activities, 2, determine consistent and appropriate avoidance, minimization, or mitigation requirements, and additionally, scientific resources that inform how we understand impacts from those in-water works to kelp that are identified, evaluated, and potentially mitigated for are lacking. And so what we need in terms of bringing this back to the science community is an understanding of the lens from those doing those rules and regulations, that perspective of what can they use and point to from science to justify how they apply asking people to avoid, minimize, and mitigate work, or even understand what are the actual impacts of this specific project on kelp at the site before we even look at cumulative impacts. And so from this, what we're starting to draft, and this is still in a kind of vetting by our advisory group process in terms of fine-tuning language, what agencies can start tackling what, is that there's generally guidance documents, training programs, and science and research gaps that have been identified from the regulatory standpoint that I would like to share those results with everyone on this committee. And so sorry in advance, these are just where I get to the wordy slides. They're not necessarily beautiful anymore. But I do want to point out what some of the guidance document needs are. And that includes things that start out fairly simple. A Kelp 101 document or fact sheet. For those, imagine if you're reviewing a project and kelp is just one component of the ecosystem that you have to assess and think about when you're looking at a specific parcel and a site, and you only have maybe 10 minutes. And so a one-pager, a simple fact sheet on what is kelp, why is it important, why do we care about what you do to protect it, and what stressors typically impact it, can be a really powerful tool. That's something that's lacking, and it sounds like a pretty straightforward step to me to get going. Additionally, coming up with some of those, starting to address some of those key problems that I highlighted are outlines and guidance or best practices for how to collect and review whether kelp, an assessment of whether kelp is even present at the site. Again, it seems fairly straightforward, but none of this has been compiled in a way that we can really assist those implementing those rules and regulations. So what projects need a survey for kelp to verify whether kelp is present? And what goes into that survey? What makes it so that the report that's come out the other end is actually suitable and usable? Did they use appropriate methods? What are the methods that are appropriate for verifying if kelp is present at a site? What other resources and web maps can you use to cross-check whether that survey also flagged whether there was kelp there? Did they do it in the right season? Is there, annual variation that might need to be taken into account? And if there's a site visit done, what other kind of considerations can you have? Appropriate tide window, again, time of year. Can you really see kelp if you're standing on the shore and looking for something subtidal? I don't know, but again, outlining all of these things would be a tool that'd be very valuable. Another thing is reviewing a habitat impact assessment. So if we've determined that kelp is at this site, how do we know if what's being proposed is going to impact kelp? How can we point to it? A summary of stressors that can impact kelp and at what scale. So considering some of those physical impacts to kelp, reduced light, physical disturbance and sediment, water temperature. And how does that really apply at the site scale? Also, a very important step is acknowledging the uncertainties and knowledge gaps we have, and an overview of impacts to kelp associated with kind of a number of shoreline projects that we know happen in Puget Sound or Washington State or Salish Sea, whatever you have. We've got a number of examples of questions that came up often, which are docks, mooring buoys, marinas, temporary barges that are often used in construction, and bulkheads. And so these are kind of very small scale questions that we've had, and what we've heard from reviewers over and over again, they need numbers, very specific numbers, and that goes into how they actually apply a mitigation sequence for those projects. So they first look at how do they avoid impacts to kelp and the other ecosystem components they're trying to protect at a site. And so buffer distances are often pointed to as a number. This is often 25 feet is kind of the default number of cases, but appropriate buffer distances for kelp to avoid impacts from all phases of an activity. So construction of whatever's happening and ongoing operation and use. And then you move to the next level, which is what are appropriate design standards, other construction strategies or considerations that minimize impacts to kelp. For docks, a lot of this points to things like for eelgrass, which is orientation of the dock, width of the dock, elevation of the dock. Next, we come to appropriate strategies to quantify a loss of habitat function. So if you can't really avoid or minimize, how do you even quantify what sort of acreage or compensation has to happen elsewhere? And then we get to appropriate compensatory mitigation. So what kind of in-kind mitigation can happen for kelp if it is being displaced? What defines kind of success? And what are monitoring options and durations that are needed to assess whether it worked or not? Did you actually compensate for the impact you had to kelp? And what I've heard a lot too is, when is there, how do we support and what qualifies as a no altogether? Are there activities or specific kelp habitats where you cannot mitigate for the anticipated impacts? What actually scientifically can support those in a regulatory position to say, no, from what we can see, you can't avoid and you can't compensate for this? So big, heavy loaded questions. We also, this is not at all comprehensive, but this continues with the site scale theme of capturing research and knowledge gaps that came up often. Just to relay back to this work group, in terms of what questions we heard over and over, which are things like temporary construction impacts versus how you approach cumulative impacts over long term from permanent structures. Another is impacts to benthic kelp versus floating kelp from overwater structures and temporary barges. Sorry. Another one that I can put a link to in the chat when I'm done talking is that Fish and Wildlife put together a great small overwater structures review, and Max Lambert captured a lot of great research questions specifically around small overwater structures and kelp. And so we just kind of point to that to lump in a lot of our overwater structure questions. Best practices for successful compensatory mitigation and how to monitor or assess if that mitigation was successful. How to conduct, and then this is a big one, is conduct a review of existing projects to determine impacts to kelp or efficacy of the mitigation measures that were employed. Doesn't this sound like really fun research to do? Sorry, my throat is dying. And then lastly, create some sort of list of kelp research to track what is being done, and including not what's been done, but also what's in progress. And so with that, I'm going to stop sharing and just say, I know this was a lot. This will be available eventually on our website with the kelp plan once we've got a little bit more steps finalized. And the next steps with our work group are to continue to kind of relay those back to the agencies that we're working with and start mapping out who can take the lead on what to start addressing some of those recommendations. So with that, I will stop talking and I'm happy to answer questions.
All right, so feel free, if you do have a question, we have a few minutes left. You can use your raised hand function or you can enter it into the chat. We're a pretty informal group. We have one hand, let's see, Lisa.
Yeah, I am aware of like an environmental calculator for the nearshore that NOAA, I believe, is coming up with. And I wonder if there's any way to kind of, you know, sneak some kelp, you know, quantifications in there somehow, or use that tool instead of, you know, recreating a wheel. Just a thought.
Definitely. Yeah, that, and I know Dave's on the line and maybe can speak to that more. We've tried to kind of coordinate with some of the NOAA people, which I think are hopefully considering kelp. I think the question is, and maybe Dave can answer this, are there knowledge gaps that they have in terms of even supporting what they put in for kelp in that nearshore habitat calculator?
Yeah, and I'll add to that. Yeah, Stephanie Anger is the one that's leading that at the Lacey office for NIMS. And, you know, there's many people involved in that project, but she's kind of the main point of contact. And she is trying to factor in kelp to the degree that she can on a species specific basis, and to a broader degree just as part of the SAV community. So, where data are available, she's trying to incorporate some sort of a specific metric or, specific elements that can be incorporated into valuation of the habitat, but it's not always possible. And, she's always looking to improve that tool and that mechanism. So certainly worth having that discussion, and we have been in touch with her.
Okay. We are at time, so I would recommend if you do have any additional follow-up questions for Dana, you can use the chat and we can keep an eye on that as well. Okay. Anything else, Dana, before we move on to the next? Okay, sounds good. All right, so we are going to move to Nick now with NOAA to talk about changes in kelp forest communities off Washington during the 2014-2016 marine heat wave. Nick, looks like you've got your slides ready to go.
Yep. Do they look? You should see the presenter view, or the slideshow view, which is
It looks
the slide.
great. It is.
Okay,
Looks like
excellent.
the right. Yeah.
One never knows. We don't get to use Zoom very much, so we're
Welcome.
all Google Meets. Yeah. Okay. So this paper just came out in MEPS. As the title suggests, it's about changes in kelp forest communities off Washington during and after the marine heat wave and in response to sea star wasting disease. And so there's a bunch of us involved in this, and other people driving boats, and the authors are there. I think Kelly's here and Helen's obviously here. And I don't know if anyone else has showed up. But this is some dive surveys that we've been doing since about 2015. And we had originally started them for other reasons. We were partly interested in the impact of sea otter restorations on kelp forest communities, and there was a paper by Ole Shelton back in, I think, 2018 on that. But we've continued so we started doing these surveys kind of to repeat some previous surveys, and then we sort of shifted them a little bit and continued on. And then I think we're all aware that there have been these two sort of major events that have impacted kelp forest communities along the West Coast in general. There was the marine heat wave in 2014 to 2016, when there were very large die-off of kelp's forest in some areas along the coast, especially like Northern California, but not in others. And then obviously sea star wasting syndrome with coast-wide die-offs of potentially important mesopredator. So there's been a lot of research in other areas on kelp forests, and here I'm thinking, more along the lines of dive surveys in kelp forest versus obviously people like Helen and Kathy Pfister have done kelp forest things, kelp canopy things, but less sort of dive surveys in kelp forests. So we figured, part of the point of this paper is to fill in that gap in relation to what happened along the Washington coast during this period, and just sort of kelp forest ecology in general along Washington. And so we had sort of four sort of major goals. One was we wanted to look at the prevalence and timing of marine heat waves and sort of warm temperatures along the coast, because the impacts are not of the heat wave are not necessarily consistent across the entire coast. And we wanted to ask if and how kelp forest communities changed during that period. Now, we don't have before data, so we're really just looking at what happens during this period. We have canopy data from before, but not the dive data, and I'll talk about more of that in a second. And then we were also sort of interested in whether the community composition of kelps, invertebrates, and fishes was structured more by sort of differences in space, sort of site-specific things, versus temporal variation. I'll show you what I mean by that a little bit. And then we had two sort of more specific questions. Kelp forests, a lot of research in kelp forests focuses on top-down effects of things like trophic cascades and the urchin herbivory on kelp themselves, and then sort of what you might think of more bottom-up effects of, say, kelp forest being essential fish habitat for fish recruitment. So I'm going to do sort of two little side diversions along those topics as well. So we sampled 5 sites along the Washington coast, mostly pretty much within the Olympic Coast National Marine Sanctuary, and they're shown here. So Nia Bay, Tatoosh, Cape Lava, Cape Johnson, and Destruction. Nia Bay is obviously in the Straits, but it's still kind of out there. And we do a pretty standard dive survey. There's sort of at each site, we have 2 locations where we drop that are, you know, at least 100 meters apart, a couple 100 meters apart. And then we just do a bunch of dive transects at 5 and 10 meters depth, where we're, you know, counting and estimating sizes of fish and sort of larger and macroinvertebrates. And we also count kelp stipes. These are mostly Macrocystis nereocystis, misspelled, and Pterogophora. I did this yesterday in about 20 minutes because we're in the middle of this huge ecosystem status report, and I've got these models that weren't converging. Those are the main things. We do, obviously there's other kelps down there, but those are the 3 sort of really sort of dominant ones. Macrocystis nereocystis are obviously canopy, and Pterogophora's sort of sub canopy, you know, sort of 1.5 meters above. And we have a time series that runs from 2015 to 2022, although we missed 2020 for obvious reasons. And then on top of that, we added, we have the kelp canopy data from Helen and DNR, I guess. So first, if we just look at the sort of big picture of what happened, both in terms of temperature and canopy cover, the top graph there is showing sea surface temperature, sort of average, both at individual sites and average across a bunch of sites. And 2013 was actually hotter than 2014 in terms of the mean monthly or the maximum monthly temperature in a given year. But 2014 to 2016 was warm, certainly warmer than the previous sort of, say, 5 or 6 years. And then it cooled back down and then got hot again in 2019. And in response to that, though, we did see about 50% drop in the cover of canopy kelps, but really by 2015 or by the end of the time series, they recovered pretty quickly. So it was 2013, 2014, pretty warm, canopy cover drops by about half. But then it pretty much just came back. In other areas like, you know, Northern California, they were having, you know, 98% drops in canopy cover or complete eradication. So much stronger impacts in other areas, and certainly a brief but not persistent impact at our sites at least. And this is canopy cover for the sort of associated with those areas. In terms of the marine heat wave, I'm going to come back to this again later, but if the marine heat wave occurred in 2014, we went like, why was 2013 then so warm? And the graphs on the right are showing a bunch of marine heat wave statistics. So it's, let's see, there's a sort of seasonal average, which is the blue line. And then I think that the red line is a sort of 90% threshold where the temperature is above sort of the average of the last, I think it's like 30 years. And then the little filled in red parts are the sort of identified marine heat waves. And one thing you can see is that there was more marine heat wave activity in the sort of 2014 to 2016 period, but it actually occurred in cooler parts of the year than the warmer temperatures in 2013. So like in 2014, and in particular, it's sort of like in the spring and later in the fall. So they're warmer than normal, but they're not. Marine heat waves are defined by anomalies, and so they're not necessarily hotter than, say, species tolerances, and I'll come back to that. Whereas if you look at the 2013 data, there aren't really as many heat waves. There's certainly more sort of activity farther south near Destruction Island, but those heat waves are actually during the hottest time of year. So it got much hotter. And so the actual absolute temperature was warmer in 2013. So then one of the things we did was just to look at the sort of trends in major focal groups, fishes, kelps, sea stars, and then macroinvertebrates, and then rockfish young of year. And here the kelps are kelp stipe counts. So we're counting not canopy cover, but just actual density of stipes on the bottom. And remember, we don't have any before data, so we don't have the 2013 and 2013 stipe counts. But basically, they were a bit lower in 2015, and then pretty much all species increased from over the time period and then sort of stabilized a bit. So we saw the canopy cover come back pretty quickly, and we see the stipe density also coming back from 2015 on. we saw a huge increase in purple urchins, mostly at Tatoosh Island. This is a 164-fold increase in purple urchins. And then they dropped off a bit, but big increase in purple urchins. And we didn't see an effect on the kelp really, although I'm going to come back to that in a little bit. The other thing people are obviously interested in sea stars. We didn't see that many. We saw only, in particular, we saw only 8 Pycnopodia over the entire time series and none in the last few years, which obviously isn't surprising given the die-off, but there's no real evidence of recovery there for those. There's some groups like brooding stars that did sort of start to come up, and there were always kind of blood stars and leather stars around. Most of the other ones were pretty low density. Pisaster, it's subtidally, didn't really see very many at all. And a few poking around. So not a lot of evidence that there's been sort of like a big recovery of sea stars or anything like that. Obviously, we didn't catch the decline because it happened sort of 2013, 2014 before we got there. But we don't see any recovery at this point, at least at our sites. For the fish, this is sort of a subset of the fish we count, the ones that are kind of abundant enough in the counts to mean anything. Mostly the fish communities were pretty stable over this time period, so they didn't really respond to any changes in kelp cover. Except for the black rockfish, which is the black line, which actually which did increase over the time period from about 2015 up to 2019 and then dropped off a little bit. That is probably due to an extremely large recruitment of black rockfish in 2016, which you can see in that bottom panel. So that it's YTB is yellowtails and blacks. They're kind of hard to tell apart when they're only 2 centimeters long. So there was a big burst of recruitment of those species, yellowtails, or excuse me, a big burst of black rockfish, maybe yellowtails, that seems to have resulted in an increase in larger black rockfish at that site, sort of adults, which is nice that you have recruitment leading to increases in adult abundance. So stepping back a little bit, I sort of mentioned this before, and this might be a little out of order, but if it got hot, why didn't the Washington coast lose more kelp initially? And this goes back to, I think, a difference between absolute temperature and temperature anomalies. So the marine heat waves are defined by anomalies. The kelp probably cares about absolute temperature in terms of its thermal tolerances. So, and if we look at other areas along the coast, we did have marine heat waves during that period, but the temperature never got as hot as it got in Northern California, where it reached a monthly maximum of around 16 degrees, which is above this not particularly well-defined threshold of about 15 degrees, where both Nereocystis and Macrocystis growth kind of dies off, drops down. And Southern Oregon also did not see big decreases in kelp cover, and some even saw increases in some places. And for both us and for Oregon, it just never got as hot as it got in an absolute sense as it got in Northern California. So that's probably the main reason why we didn't see these huge, like 90% decreases in kelp covers. It just didn't go over the thermal tolerances. I'm going to come back to urchins and kelp in a minute. The other thing we were just looking at was we did some multivariate analyses to look at community structure. And really here, partly the idea is to look at the effects of both space and time on structure and community. So like, do communities change together over time, or are sites like just basically always different from each other? And really most of the variability in community structure is made up in variability among sites for kelp, invertebrates, and the fishes. So that's those, if you look at the ordination plots, these are from a canonical analysis of principal coordinates, was just a constrained type of ordination. And the points are colored by site, and you can see there's, for the top 3, there's pretty consistent differences among sites. They just don't overlap from year to year. So community structure is pretty stable at these sites through time. Sites remain different from each other. The difference is obviously the rockfish recruits, which we might expect to be, based on what we know about marine recruitment, we'd expect that to be more temporally and spatially variable, and that's a bit more of a mess. Another way to look at it is the graph on the right, which is looking at the variance explained by each of those terms. And what you can see is that for the kelp, the inverts, and the fish, most of the variance that is explained by the site term. There's a lot of unexplained variance in this residual term all the way on the right. And then kelp also, some of it's explained by depth and site times depth, which you'd expect because it's of its light requirements. The difference, and then most of the variation in juvenile rockfishes, which is the kind of yellowish bar, is explained by this year term, which is just probably mostly due to that big recruitment for black rockfish in 2016. But recruitment varies in space and time, so it's not particularly I mean, that's what we would expect. One of the things we were interested in is top-down effects that urchins might exert on the kelp, right? So we had this, and well, two things. One, this graph, this is a bunch of graphs that are different spatial scales, and so they can be kind of confusing. The main takeaway is that for the most part, we didn't see a lot of evidence that increases in urchins led to any kind of decrease in kelp. And that's the top graph is just averaged across all the sites on the entire coast by year. The bottom two graphs are site by year. And there's not really anything there that there's one that looks like is a bit of a exponential decline, but that's largely because there's sort of no urchins at the sites that tend to have macrocystis, because most of the urchins were at either destruction or tattoo shell. It's a bit of an artifact of just the distribution of things. And then there's some, even then, for example, the bottom, their pane F, there actually appears within the site of Nia Bay, it's sort of like averaging transits across a year. There was actually a positive correlation. So basically, didn't seem to be much evidence for a top-down effect of urchins. At Tatoosh, where there were a lot of urchins and near assisted, there's a little bit of evidence that at the transect scale, there was a negative correlation, which we would probably interpret it as urchins causing sort of patchy distributions of kelp, but not really reducing the entire canopy in a huge way that we saw any of the canopy data or the total stipe counts. So it's creating patches, but it's not as it's not a huge effect. And we want to ask why this is happening. One is that I'm not putting up the numbers here, but the urchin densities were actually not that high. Even when they got really high, they were still well below the densities that associated with the big die-offs in Northern California. Really only maybe a fifth or a half as high as those. So urchin densities were really low, so there was a 164-fold increase at Tatoosh Island, but it still really weren't that many urchins compared to some places where we've seen the extirpation of kelp. And some of this is probably relates to urchin behavior, because urchins can feed in two ways. They can be passive detritivores, just kind of sitting around and eating drift kelp. And we never really lost that much kelp, so there's still probably a lot of drift kelp around for them to eat. So they may not have changed their foraging behavior into becoming active herbivores. And when they are active herbivores, that's when they tend to wipe out the kelp stands. And some of this probably relates to habitat complexity, which Zach can tell you all about. And Tatoosh is pretty complex habitat, so it might retain the kelp in there, the drift kelp, better than you would expect otherwise. And then there's some fancy math that we did, which is, I already said that. The other thing we checked with, and we're almost at the end, is we looked at the relationship between kelp abundance and in here, particularly canopy cover, and or stipes, number of stipes, and rockfish recruitment. And this is done in a two-part hurdle model. So you sort of model presence or absence of, and then you, in a separate way, you model abundance of the positive observations. And so you get sort of different parts to the model that come out. And one is that like the modeling shows that like when you have more kelp, you are more likely to get rockfish recruits along that transect, right? So kelp, more likely to get rockfish recruits, but we didn't see an abundance effect. And that is probably just because of the, you know, you get more recruits, you're more likely to get the recruits when there is kelp, but there's a lot of spatial and temporal variation and other things like larval supply and things like that that could obscure the abundance trends. And this survey was also not specifically designed for this. So you know, you might be able to do a better sort of recruit sampling in a different way to better define that. But either way, in an essential fish habitat sort of way, you know, the kelp, as we might expect from work in California, in other areas, kelp is good for fish recruitment. And then, so just including, so Washington did get warm, but mostly in 2013. And there was marine heat wave activity on the coast, but it was just not as warm, which as other areas that lost a lot of kelp. And we saw pretty stable communities over the time, even in areas where we had these large increases in urchin abundance, especially the purple urchins. And I will stop there and take any questions.
Okay, thank you, Nick.
Yep.
So we have about 4 minutes for questions, so feel free to raise your hand or use the chat.
And Kelly should feel free to, if he's still here, to chime in if he has any.
Okay, we have a question from
Answers.
Helen and then Jenny.
Yep.
So I'd just like to raise a distinction for and really to this emphasize why it's so important for us to look at the coast as well as Puget Sound. As Nick is saying here, that the kelp communities were largely stable on the outer coast, but during this time period, we saw huge losses within Puget Sound, and many of those areas haven't recovered. I'm thinking of the inner basins like South Puget Sound, Central Puget Sound, sites in Saratoga, Whidbey that the MRCs have monitored. And so this is one of the really powerful things about this work for me is, and thank you, Nick, for making those points really clear about the difference between temperature anomalies and absolute thresholds. Because I think, you know, to me, when I look at this work, that's one of the big things that are clarified for me, and I really appreciate it.
Jenny and then Chris.
Yep.
Jenny, if you are speaking, we can't hear you. I've lost her, so we're going to go to Chris.
Hi, great, thank you. Hello, everyone. Nick, great presentation. So I'm Chris Neufeld with the Kelp Rescue Initiative and Banfield Marine Sciences Center up here in BC. And we've done some work recently just looking at some oblique aerial imagery, so a very kind of coarse scale look at how kelp forests have changed on the outer coast and inland waters kind of throughout BC in some little pockets where we had some data. And we found that they're in places where the water is regionally warm, as you say, not heat wave specific, but actually just beyond thermal tolerances. For sure, we've seen some declines in both species, but also for sea urchins, we've seen most of the declines on the kind of even if it's the kind of outer coast, it's on the inside of these large islands where it's more wave protected. Mm-hmm. I'm just curious, you know, waves can often push urchins down from that kind of at least first few meters of habitat in those more wave exposed places. Do you see any patterns with urchins at the site level, for instance, where you are seeing less kelp and more urchins in places with bigger waves, basically?
Well, for these dive sites are kind of chosen to be diveable. So there's, and I think in the 2018 paper, Oli, and they'd originally done some wave exposure type stuff for it. There's not a huge difference in the wave exposure for the sites. And, you know, like there's lots of reasons why you might have, you know, and Kathy can probably go on for ages about this, but wave exposure certainly matters for kelp, among other things, and nutrients and all that kind of stuff. These sites don't have huge differences in wave exposure. Like the Tattoo site is on the inside. A lot of the other ones are sort of on the more protected side, so we can't really track that. There are certainly differences in both kelp and urchins with depth, which I can dig out somewhere. I have it's in the appendix of that paper. They're just plotted there, but there's not, in some areas there's just like isn't that much kelp at 30 feet versus 5. So it's
Sure.
Anyway, so it's sort of I'm not sure if I've actually answered the question, but
No, that's good. I mean, I appreciate it. Just more, I'm just thinking sort of, when we look at these certain spatial scales, like your data clearly shows things are pretty stable in these regions. And at other spatial scales between, Washington and Oregon and California, we're seeing all these other patterns, and we're seeing those same kind of patterns all the way extending up into BC. Just kind of interesting to think about the different scales that these different drivers might be acting on.
Yeah, I mean, the paper has a section on kind of, there are large differences in, the number, and somebody pointed out otters in the chat. There's like differences in where there are otters. Like there aren't really otters in the North Coast. And then if you have no otters in North California, if you don't have otters and you don't have Pycnopodia, then maybe you have nothing to keep urchins intact. Where we do have otters, although they tend to be farther south along our sites than they are. At least I haven't checked their numbers recently, but in the Ole Shelton's 2018 paper has a whole thing on the distribution of otters over time for people interested. They tend to be, they sort of showed up and then they move farther south along those sites. They're sort of
Right.
more down.
Great, thanks.
Yep.
All right, we are at time. So any last words, Nick, before we move on?
No, it's all good. Call me, email me if you have questions.
Great. Okay, thank you very much. All right, we are going to move on now to Danielle with DNR. She's going to talk about the floating kelp vital sign indicator, as well as highlights from the 2022 aerial and drone imagery collection.
Great, thanks, Elizabeth. Let me share my screen. Whoop.
That's presenter mode.
Thank you.
Perfect.
All right. Hi, everybody. I'm really excited to talk with you today about our nearly complete floating kelp indicator for Washington State, and some of our next steps for development and monitoring. And for the floating kelp indicator, as many of you know, we're a really diverse project team with representatives from the Samish Indian Nation, Marine Agronomics, the Northwest Straits Commission, the University of Washington, Sea Grant Washington, and DNR. And in the first section of my talk today, I'll introduce you to the first edition of the indicator. And it's really the last step in the development process, but the first step toward building and using our floating kelp indicator data. And then in the second section of the talk, I'll give you a sneak peek of some of the aerial imagery that we've collected in 2022. Excuse me. My description of the floating kelp indicator today will draw from the draft documents that are currently out for review. You can access the products through the graphical abstract, and Helen either has or will add a link to that in the chat for folks to take a look at that. And the products that we have for the floating kelp indicator fall into two different categories. The first is preliminary results, which include a statewide summary report presenting findings, including the statewide indicator. And we'll dig into that a bit later. And secondly, the interactive map, which explores the data, and I'll give you a peek as to how we've organized that as well. And then second are supporting documents which hold more detail. So we have a program design and data assessment protocol, and as well as sub-basin reports. And so these really dig into those fine details, all of the different data sources and how they were incorporated. So if you want to take a deep dive, those are great to look at. And if you're wondering where the Puget Sound vital sign for floating kelp canopies has gone, that is part of our products for this indicator. And so this indicator includes information to inform the floating kelp vital sign. And so if we zoom out and consider a broader context of research, monitoring, and management of kelp in Washington State, the indicator project draws from, but also informs, a broad array of kelp-related work. Potentially notable for this group in particular are habitat usage, stressor research, and field expeditions. And the indicator really serves as a nexus for data of kelp status and kelp knowledge of many forms, as well as providing insight into data gaps, research needs, and management issues moving forward. And so as our team was developing the floating kelp indicator, we approached the design with some key considerations. And these considerations have been refined by our project team and by input from workshop participants and other folks. And so you can find this table that I'm going to show you and related discussion in the Monitoring Program Design and Data Assessment Protocols document. And so we wanted to be able to inform diverse audiences and produce an intuitive summary for rapid communications. But we also wanted to then go from that really broad scale information down to detailed products where you can look at the data itself. We wanted to consider both short and long-term trends in floating kelp abundance. And we wanted to divide that up into subbasins within Puget Sound, as well as smaller assessment units. So the reason we're doing this is because we anticipate there will be some areas which are doing better, some which areas are doing worse, and we want to be able to see how that varies across the state. And as we changed the name at the beginning of this meeting today, this includes the Open Coast as well as Puget Sound. And we really wanted to be able to maximize the use of available data while also considering and taking account of data limitations. And so initial data sets that we're including are canopy and bed perimeter of DNR MRC volunteers in the Samish Indian Nation, and then other available data are included as we have access to them. And I'll talk about this more later, but if you have a look at the indicator and you realize important data are missing that you either have access to or know of, we would love to hear that to incorporate that into the next iteration of the indicator. And we wanted this program to be scalable and able to develop a strategic plan for looking at what research needs we have into the future. And the indicator itself includes monitoring, which is limited to describing status and trends, but we wanted to make it possible to consider linkages to stressors, management actions, and other ecosystem components in human well-being. So with all of those considerations in our mind, our project team went to work on developing a floating kelp indicator to best incorporate as many of these considerations as possible. We came up with a two-tiered product that assesses long-term trends in floating kelp area at a local scale, as well as an integrated assessment at a sub-basin scale that synthesizes hard data with other sources of knowledge to determine floating kelp status. I'll go into detail on both of those here now. First, the indicator tracks the portion of kelp forest that floats on the surface. And we chose this metric because programs throughout the world monitor the extent of kelp forest floating at the surface. It's also possible to measure using a number of different methods. So you can observe floating kelp canopies using drones, fixed wing imagery, and kayak monitoring, which allows us to collate as much quantitative data as possible to assess trends. For the indicator specifically, bed area is the primary metric. And this is defined as the area encompassing kelp tissues floating on the surface and the small gaps between adjacent individuals. And we picked this as the most data-rich metric because we have bed area monitoring data from the most locations across Washington. We also use secondary metrics like canopy area when those data are available. So here you can see the red is the canopy area and the yellow indicates the bed area. Excuse me, we found that in general, over time, canopy area and bed area are pretty well correlated. at locations. So using this definition, we're able to assess long-term trends in bed area at 172 locations using linear regression. We also assessed 5-year recent trends, but due to the natural high year-to-year variability in kelp abundance, the indicator emphasizes long-term trends. And results from these regression analyses help place each location into one of 6 trend categories. So the top 3, increasing, no trend, and decreasing, are pulled from the regression. Increasing means a significant positive direction, decreasing, significant negative, and no trend is no significant change over time. We've also added in a category for total loss, where kelp has been present in the past, but was not present in the most recent year. And finally, we have limited data, which means that quantitative data are available, but they're not suitable for regression analysis for one reason or another. And then also areas where there is no floating kelp due to the environment. And we can look at trends at specific locations on a map, or we can summarize the % of locations in each trend category to understand broad patterns. So as I mentioned a bit earlier, we summarize these trends within 11 different subbasins. And these are commonly used oceanographic units, which are also generally fairly familiar to folks in Washington. And you can see that there are differences in their trajectories. For example, the North Coast, it tends to be doing well, with most locations either increasing or having no trend in bed area. And areas like the South Puget Sound, where it's dominated by locations with declines and total losses. So this is a lot of data, especially when you look at each and every subbasin, and it takes a while to digest it. But one very strong message we heard throughout the development of this project is that we need some sort of summary that's intuitive and rapidly understandable. So let's get to that. In order to address that, the project team developed Subbasin Status, which provides a rapid summary of the state of knowledge within each of the subbasins. Since we're talking about numbers today, I'm focusing on our quantitative results, but I want to emphasize the fact that these subbasin status assessments are based on multiple ways of knowing. So this includes Indigenous scientific knowledge, local community reports, and historical ecology studies. Without those sources of data, a lot of these assessments would have still been insufficient data. And excuse me, as a note, often status is assessed relative to a defined baseline or target, but floating kelp doesn't have either of those metrics yet. So we define status based on considering the trends in the resource along with the data certainty for each of those subbasins. The categories are stable, concern of decline, insufficient data, and no floating kelp. So the difference between concern of decline and decline is that decline indicates a substantial decline, whereas concern of decline means that we have some information to suggest there may be declines, but we don't have enough quantitative information yet in order to classify it within that substantial decline category. And notice that there are really stark differences in our assessments of subbasins across the state, but also that there's quite a bit of missing data. Blue indicates data gaps that are large enough to preclude any subbasin status assessment. So that is North Puget Sound and Admiralty Inlet. And yellow indicates that there are some data that indicate decline, but we need more data in order to confirm or revise that assessment. Furthermore, data completeness and coverage is not even across subbasins either. And so there's a different number of locations monitored, as well as a variation in the % of nearshore extent monitored. So On one end, we have Admiralty Inlet with only 2 locations monitored, encompassing only about 1% of the shoreline, as opposed to areas such as the northern coast, where we have 20 locations and 100% of the nearshore extent monitored. So this leads us to suggest that comparing results amongst subbasins must be done with caution, given this data variability. And here's another way to look at the data that we do have. So this plot shows how many years of data have been collected at locations in different subbasins. You'll notice that the distribution comes in 2 different forms. There are multiple sites with a lot of data, with 30 years of data, and a few sites with shorter data records. The exception is the San Juan Islands, which has many locations with 3 years of data, but this needs to be developed further as methodological data limitations can currently preclude regression analysis. So I'll come back to these data gaps in a few minutes, but I'd first like to show you one way that we've worked towards making the indicator accessible and understandable by developing an interactive map. It allows general users to view the indicator at a statewide scale, but it also provides location-specific information for each of the 172 locations that are currently located within the indicator. So let's dive in. When you first enter the site, a splash screen shows up with summary instructions. Most people tend to just push OK so that this goes away as quickly as possible and they can get to the map. If you do that and need to come back to it, it's possible to use the information button, which I'll show you in a minute, to get that back. When you open the map, it opens to a statewide view, and it shows you the subbasin polygons, which are color-coded by status, which we just discussed. And there's a legend to the right-hand side. Each of the points show the long-term trends at each location. And you can see that the colors are defined here to the right. So let's explore a bit. If you zoom in with your cursor, you can click on a subbasin polygon in order to get more information about that. So say you're interested in the eastern strait of Juan de Fuca. If you click on that polygon, it gets highlighted in blue and a window pops up with information about the subbasin, showing you the status, the name, and how many sites are, what the site trend summary is. So if you want to know even more, you can go ahead and click on that thumbnail. And a one-page subbasin summary opens, which has a location map, key findings about that subbasin, bar charts showing the long-term and short-term trends, and then a summary of what's happening within that subbasin. And if we go back to the interactive map, if you zoom in, the colored subbasin polygons will disappear, and you can see gray polygons that show the boundaries of each monitoring location. And floating kelp polygons also appear. The blue shows the maximum extent of floating kelp across all years surveyed, and the yellow shows the bed location during the most recent year. So you can sort of see those summary polygons for your particular location you may be interested in. If you want to know about a specific site, you can click any point to find more about that location. So if you click here on Evie's Landing, you get a pop-up which shows you that it's increasing. There's 7 years of data, and it shows a thumbnail of the bed area over time, which gives you a glance. But if you go ahead and click on that, another one-page summary pops up, and that includes the summary of the results for the site, the bed area by year, the map, as well as a photo if we have it, and links to various websites where the data come from. And this example is part of the MRC work, and each type of data has a different type of one-page summary. So one last note, the icons in the upper right-hand corner are the widgets. So if you got rid of that splash screen and you want it back, you can click the eye. If you want to change the base map, you can click the square. If you want to switch which layers you can see, you can click the stack. And the one on the left shows you the legend. And so I hope that everyone will have a chance to take a look at the interactive map. So please let us know if you have any recommendations for changes or enhancements that we can make to the next iteration. So as we've said, the indicator is composed of this annual bed area on long-term trends, but other forms of information are synthesized into it. And this slide has a lot of text, but basically this is considering where we can include data, how that data can be included into the indicator, and what would be needed to move forward with those data. So we've talked about the data we have, the ways we've visualized it so far, and the fact that large gaps remain across much of Washington's waters. So you can see here all of this missing data and all of the areas where we need more information in order to determine status and trends of kelp beds. And so one of the ways that we've started to address these gaps is to up our aerial surveys to cover a large portion of the state. In 2022, we collected both drone and fixed wing imagery. For the drone imagery, we conducted 18 surveys at 10 different sites, and about 200 hectares were surveyed. And we also flew 7 sites twice during the same low tide to see differences in visualization of the canopy at different tidal heights on the same day. And in 2022, we also acquired 4-band 15-centimeter resolution aerial imagery using a small fixed-wing aircraft across all areas of interest shown here. The goal was to collect this imagery at low tide, slack current, and good conditions, although there's a few locations that were surveyed later in the season that we had to stretch our requirements a bit. We'll be looking into how that affected visualization of kelp during our analysis. The fixed wing imagery ended up having 760,000 acres surveyed, which includes about 2,000 kilometers of coastline in Washington. So this is very exciting. It will give us a really nice snapshot of kelp in the state, across much of the state. And you can see here all of the areas where we do have imagery. And of course, we're zoomed out, so you can't see the individual kelp areas, but if we zoom in, you can see Admiralty Inlet. And we're using this, and we can zoom in even farther. And we can see a kelp bed emerge just off the coast. This is shown in false color. And given the abundance of the other types of algae and other false returns in the images, so things like the warm shoreline, rocks, land plants, the first step that we're working on is to create a mask around the areas that we expect will be floating kelp. So here's an example of the first pass of this masking in Admiralty Inlet. And our goal is to include all areas that may be kelp and exclude any spurious areas. We're currently working with a masking buffer of 10 meters around the edge of the bed. And like I said, this is just a first pass at this one, and we'll be working as a team to mask the state. And here's another example from Possession Point. And so we'll be masking across the state, and we anticipate that this will be time consuming this year, but that we can then use this mask to form the basis of masking for future years. So we may make small changes to the mask in future years, but we'll have an idea of where kelp is likely to be. Stay tuned for more analysis methods and results soon, and thank you very much.
Thank you, Danielle. Okay, so we have maybe time for one question. And I'm going to actually kick it to Celine, who says, what imagery do you use for masking?
We are using our aerial imagery for the masking. So that's that 15-centimeter resolution imagery. And that we will be doing the same imagery that we'll be doing the analysis on.
Okay. And I think we have time for one more question from Jenna, and it says, does DNR plan to do repeated drone aerial imagery collection?
Yes, that it is dependent on funding. What we're able to do each year. We're currently working on a plan, but the hope is that we can continue to collect both aerial and drone imagery as we move into the future. How much of that we collect will depend on budget and constraints, but we're excited to develop this further.
Okay. And let's see, I think we are at time. We are getting some questions in the chat though, so I might direct you that way and we can respond in the chat.
Yep,
Just
sure
that we
thing.
can, great, okay. And there's some additional comments, so feel free everyone to continue the conversation over there.
Thanks.
Okay, thank you, Danielle. So we are going to move to our next presentation. This is Zach with the Seattle Aquarium, who's going to be discussing the ROV benthic survey methods he's working on in Elliott Bay.
Thank you, Elizabeth. Can you all hear me and see a title slide?
Well, I can hear you. I don't see anything yet, but it shows that it's thinking on it. Do others see a presentation yet?
Let me try something else.
Yeah, I don't see one either.
Okay.
Okay, how about now?
It says you're started. Oh, here it goes. We're getting closer. You just went black. You're back. The screen is still just a black screen.
Yikes.
This feels like a Zoom issue, maybe not a your computer issue, but I can't tell. Someone says you may need to move your presentation to a different monitor. The other option, if you'd like to quickly shoot me your slides, I could try from my machine. He actually looks frozen to me.
Sorry about that. I keep
There's
getting
two of him.
booted out. Let's try this again.
Still nothing. What we could do, we could swap. If Jamie Selleck's ready, you could shoot me your slides and I could get them loaded while Jamie presents, and then we could move back to you. I think we've lost him. All right, Jamie, are you? Oh, there he is. He's back.
Yep, sorry, it keeps booting me out. I will email you my slides, Elizabeth. Thanks for doing that.
Yeah, absolutely. Okay, Jamie, I know that you're a little ahead of schedule. Are you out there? He was sticking to the agenda. I see he's here, but he might be taking a break. Jamie, are you out there? Okay, well, I will wait for the slides then and have that ready to go. If you wanted to get started with your talking points, Zach, and then as soon as this comes through the internet, I can get it ready to go.
Yeah, I'm just attaching it. It's weird. I was sharing slides on a meeting on Teams just this morning.
Hi, this
And
is
it
Jamie.
showed
I'm
that you
sorry.
were. Oh, there's Jamie.
Yeah, I just got a text. I had stepped away for a second. I'm sorry.
No worries. We're having some technical difficulties, so we were wondering if we could move to you while we work those out.
Sure. I was about to fill my coffee, but I've got some water. That'll do.
Okay. Thank you for being flexible.
Yeah, hold on just a second. Let me bring this up. So you want me to just go ahead with my talk now?
Yeah, if you could just dive in and then we'll finish up with Zach's.
Sure. I'll take up all the time till noon. Let me share my slide then. Hold on just a second. Got it open. I just need to. Oh, almost there. All right, let's see if this works. Sorry, I missed that. Yeah, I got it. I got the text and it was like, you're being called on. Come back.
Takes a village.
All right, can you see that?
Yeah, that looks great.
All right. I didn't hear if anyone introduced me. I'm Jamie Selleck, Natural Resources Consultants. Helen asked me to present today on a report that was completed back in August of last year. A couple of you, I think, have actually seen this presentation. I've given it a couple of times. I know, like Nick, I presented to at a NOAA Habitat work group. And I'm also going to be presenting it next week at a PSEM partnership work group meeting. So we're making the rounds. But this is a big report. It's the state of the knowledge for nearshore habitat fish assemblages and invertebrates for the West Coast. You know, Helen asked if I could just kind of talk a little bit about the report, but it is massive. It's 350 pages. So I'm going to do my best to fly through the outline of the report so that you can dive in and find the information that's most relevant to you, especially with, you know, topics today like kelp and submerged aquatic vegetation. There's another component of this, the online data tools. Kate Sherman with the Pacific States Marine Fisheries Commission generally gives that portion of the talk. I included a couple of her slides, so I'll do my best to kind of review that, but there is an online data data tools website that you can access as well. So I'll just introduce who is PMAP. This was the Pacific Marine and Estuarine Fish Habitat Partnership. I know some of you are very familiar with our group, maybe not everyone though. So I'll talk about the development of the report, its layout. I'll lead you through, you know, like I said, the different chapters and sections and say, hey, you can tease apart the information that's most relevant and look through. But I'm sure most of you will all read all 350 pages because it is fascinating. I'll go through our goals and objectives, how we got at some of our ecoregions and our fish assemblages, and I'll just touch on our data gaps and stressors. I'm not going to really spend a lot of time on that, but and where you'll be able to find the report and the data tools. Acknowledgements, just important. Obviously, this was a small army to produce this report. There was a nearshore working group, science and data committee, the steering committee provided input, and then we had our funding sources from NOAA, NIFHAP, and AFWA. So some of you on the call today are actually on this list here, so thank you. So for those of you that are not familiar, NIFHAP is the National Fish Habitat Partnership. They are a national organization that acts as an umbrella for regional partnerships associated with a mission to protect, restore, and enhance fish and aquatic communities. So our regional partnership is the Pacific Marine and Estuarine Fish Habitat Partnership, PMEP, which is all of the West Coast. And our mission, similar mission, but is protect, enhance, and restore habitats within estuaries and nearshore marine environments. So there are other working groups and partnerships that focus on rivers, streams, and lakes, and we're associated with the continental West Coast. Over the course of the years since the group was formed, there's been a series of big reports produced on eelgrass restoration, state of the knowledge on West Coast estuaries, barriers to tidal connectivity. You may notice a common theme. A lot of the reports early on were definitely focused on the estuarine side of the West Coast. And so there was obviously identified need to understand the processes and connectivity for species and habitats of the marine component of our partnership. So the nearshore and offshore along the West Coast. A working group was formed in 2018. And the goal was to provide best available science to inform opportunities to conserve, protect, and restore. The report was completed in 2022, so it was quite a long process in getting a lot of the information together. This is a synthesis. It's a compilation of materials, but it's meant to put all this information as its current understanding in one repository. And like I said, there's online data tools associated with that as well. So the original 3 goals, the first 2 are italicized because those are associated to the data tools, and that was to develop a spatial data system, compile nearshore habitat spatial data sets, and then develop this state of the knowledge report. This is just a condensed version of the table of contents, but it's meant to just highlight as an outline so you kind of know how it flows through the report. The methods section is heavy on the data tool side and the classification schemes that were used to define our regions and our habitat types and assemblages. And then within each ecoregion, we identify the various depth zones and habitat types and fish assemblages. And then there's closing sections on data gaps and stressors and other considerations. What's next? What do we do next? Kind of thing. So the the hierarchy of this is that there's chapters and sections on each ecoregion along the West Coast. And then each ecoregion is divided into subregions. So here's the Salish Sea, and the subregions of the Salish Sea should be pretty obvious to you. They're the basins and the subbasins that are well defined and agreed on for different parts of the Salish Sea. Within each of these subregions, we have our depth zones. So from shore down to certain depths. And within each of these depth zones, we identify habitat types and substrate types. And then within each of those, we classify our fish assemblages and our selected invertebrates. So these are the 4 ecoregions, Salish Sea down to the Mexican border, which is Southern California Bight. The Channel Islands are split a little bit between 2 of the ecoregions. The Pacific Northwest ecoregion, the subregions you see involve like, Cape Mendocino and Northern California, and then the Oregon coast and Washington coast. And then the Salish Sea is, as you saw, the subregions there are the different basins. I'm flying through all of this. I realize that there's a lot of, I just want to kind of highlight, and we can definitely have some time for questions. Within each of these regions, we have depth zones, and the different depth zones were identified as the landward area. This is the influence of the land on the nearshore. And so this doesn't really have a distance defined. It's the top of the splash zone, the very top of the intertidal, to however far has an influence. So in some areas that might be steep cliffs with feeder bluffs. Other areas you might have very broad swaths of wetlands and watersheds that influence the nearshore. So that part varies heavily between different ecoregions. Our core zone is the bulk of this report because the core zone is captures your photic zone. So all your biotic habitats, your kelps, your submerged aquatic vegetations, your eelgrass, that's all within the core area from the top of the splash zone down to 30 meters. And then our outward depth zone is the seaward area, 30 meters to 100 meters. We didn't ignore everything beyond 100 meters because obviously in the Salish Sea alone, you've got a lot of habitats down below that. If fish assemblages were influenced by that, they might be captured within the seaward area, but that's the limits to where we categorized our state of the knowledge. And that partly has to do with the interest and the mission of PMAP and our goals. Within each of these depth zones, we identified substrate and habitat types. So consolidated and unconsolidated materials, boulder, cobble, sand, and mud. You can mix and match these. You can have sandy cobble or silty mud. And then you've got, biogenic, you've got woody debris and shell hash, and then artificial material, anthropogenic metal reefs, rubble, rock piles, the artificial habitat. But then also we have separated out our biotic habitats. So these are your faunal beds, your kelps and your seagrasses, and these are captured within different substrate types. So you can have hard bottom biotic, soft bottom biotic. I'll show some examples of that. So then the last step of that is to identify our fish assemblages within each of these habitat types, within each of these depth zones, within each of these ecoregions. And so what are the common and abundant fish species within each of these habitats and depth zones? They're common and abundant. I mean, this isn't a category of every fish species found in the Salish Sea. It's the most common and abundant species identified by their presence, their population sizes, data that's available, their occurrence within the literature. They're recognized as important common species that are established within each of these ecoregions. And so they're separated by, your bottom-oriented, your pelagic species. You've got, as I said, an example of soft bottom biotic. And then habitat generalist species that traverse different habitat types, and then ones that are solely associated with just the biotic habitats. I figured it'd probably help to just give a few quick examples of how this fish assemblages kind of came out and some of the results that we saw. We found that species richness of common and abundant species increased from the northern ecoregions to the southern ecoregions. The Southern California Bight having the most diverse ecoregion with 478 species, and the Salish Sea identified of 173 common and abundant species. We found that the number of common and abundant species in Salish Sea increased with depth. Again, we're probably familiar. We have a fjord system. We have very narrow near shores that drop down to significant depths very quickly, and so you would find that variation there. Whereas the species richness in the Pacific Northwest is very comparable. From Northern California, Oregon, and Washington, we found a lot of similarities in species assemblages for each of the subregions. If you want to get at what fish assemblages are associated, most common and abundant fish assemblages are associated with hard bottom biotic, your canopy forming kelps, for instance, you would find these listed under hard bottom biotic, and they're listed as common or uncommon by their depth zones. Again, hard bottom biotic is related probably to the photic zone, so that would be more common in the core depth zone rather than the deep seaward, which is listed here as rare in the deep seaward. So in the Salish Sea, we see a lot of rockfish species, sculpin species, greenling species associated with those hard bottom biotics. You can also look at your species richness by different subbasins or subregions. So these abbreviations at the bottom, I hope are familiar to you. The San Juan Island Archipelago, Whidbey Basin, Hood Canal, North Central Puget Sound. I assume you can see my mouse. I'm trying to circle them all at the bottom there. But yeah, you can look at how species richness varies between different subbasins and and this is produced for each of the ecoregions. What's probably also of interest to this group is how the frequency of established fishes are found on different habitat types. So we find in the Salish Sea that the common and abundant species are most frequently found on mud, sand, and rock habitat. We could also argue those are the most common habitat types, but you would find the most, the most highest frequency in species richness. But you can also look at these separated out by your different biotic habitats. So you have your seagrasses, your kelps, and your other algae, a lot of understory kelp, turf algae, things like that. All right, so that's crash course into fish assemblages. For the invertebrates, we didn't do assemblages based on habitats, although the individual invertebrates we describe have their own species descriptions where we identify what habitats they're found in. The criteria for including invertebrates was based on their level of importance to an ecoregion. So their ecosystem importance, are they a management or fisheries important species, and are they identified as population decline, so threatened and endangered. You may find similarities between species in different ecoregions, but they were selected as meeting these criteria for each ecoregion independently. But we did try to balance the number of identified important invertebrates to each ecoregion. So this is a table of your selected invertebrates, and you can find that purple sea urchins are found kind of ubiquitous. They were identified as meeting those criteria for all 4 of the ecoregions, whereas Dungeness crab was only identified as meeting the criteria for 3 of the ecoregions. That doesn't mean it's absent or not present in the 4th ecoregion. It means that it didn't meet the criteria for ecosystem importance and population decline and things like that. If you also want to kind of get a little bit about their habitat associations, you can look at their depth ranges. So we find that Dungeness and red rock crabs are fairly ubiquitous. They're found from intertidal all the way down to seaward inner shelf, whereas spot prawn is absent from intertidal, but is more common in the deeper depths. Same with the California market squid. It's predominantly found at deeper depths beyond the photic zone and outside, but it would be found in some secondary habitats. So this is where the subtidal area where you might have your biotic habitats where you might find market squid. So the last piece for the ecoregions is we invited a guest speaker to write a couple of pages on an issue that's important to each of these ecoregions. So for the Salish Sea, we identified the need to understand larval connectivity between the outer coast and the Salish Sea. And so the Pacific Northwest Crab Research Group wrote on the work that they're doing with larval Dungeness crab monitoring. For the Pacific Northwest, Steve Rumreld of Oregon Department of Fish and Wildlife wrote about disruptions in species shifts related to ecological interactions. So discussions in shifts in species ranges or population numbers associated with, say, the loss of kelp. And Central California, Dave Lowry wrote about the ecological connections between assemblages. So this is more like kind of the trophic level connection between species guilds. And then Brian Chesney, also of NOAA, wrote about in the Southern California Bight about the factors that influence that rich diversity in the southern area. So I just have a couple of slides, but I'm not going to talk about them too much, but to give you an example, we identified data gaps for invertebrates and fishes. We did find that for invertebrates, that nearshore macroinvertebrates were not as well documented historically as fishes. This isn't surprising when you look at the level of historical data for fisheries-dependent species. So fisheries-important species that we would be able to do catch reconstruction data on, or things that go back over 100 years. There's just more data available that answers some of the questions related to their historical distributions and population abundance and things like that. But obviously, has been discussed, the influence of marine heat waves and changing climate and ocean conditions, and how this is associated with sea star wasting disease and things like that. And so that's identified as a data gap that needs continued work. For fish species, some of the data gaps revolved around this question of connectivity. So just because we have similar fish assemblages between ecoregions doesn't mean they're connected from a genetic standpoint, a metapopulation standpoint. So understanding larval mortality, fish assemblage connectivity between regions, the effects on trophic cascades, that's something that needs to be a little bit better defined. We know that these species have ranges across ecoregions and they're found in different ecoregions, but how are they connected from a connectivity standpoint is not as well defined. And that would be really important for understanding these range shifts associated with changes in ecosystem habitat, similar to identified with the invertebrates in the loss of kelp beds on the outer coast, particularly Northern California and Oregon and Washington. Those are all examples of indirect stressors. Your direct stressors, obviously there's a laundry list from invasive species, shoreline armoring, aquaculture, underwater noise. I'm not going to spend any time on this, but just to highlight what we identified. Okay, so the last couple minutes, I hope I'm okay for time. I'm trying to fly through this. There's a lot of information to cover. Last couple of minutes, I wanted to show a couple of the slides. Kate Sherman normally gives this talk associated with the online data tools. And I think this is of great interest to this work group, but she's definitely the data wizard. So I'll do my best to kind of translate the information. But similar to the report for our methods and classification schemes, we used the CMEX, which is the Coastal Marine and Ecological Classification Standard. And that was used to define our substrate components, our biotic components, and setting up the biogeographic settings, the aquatic settings, and these components that went into the online data tools. So the categorization is for the online data tools is very similar to the report. And I'm just going to show a couple of slides to show some examples. So here is the Salish Sea. This is the depth zones associated within the Salish Sea. You can overlap this with habitat data. You can overlap this with fish assemblage data. You may notice that here's an example up in the Nooksack River. There's a lot of overlapping data, and this has to do with how data was defined for the intertidal zone, large areas that have broad wetlands, splash zones that aren't as well defined. You're gonna have a lot of overlapping data in those regions, but otherwise you can get some pretty good maps associated with each sub-ecoregion. If you wanted to look at the percentages, you can also pull out that data versus, so for example, for benthic macroalgae, canopy forming kelp, we can look at the, within the data set, you can look at the area coverage and the percent area along the near shore of how much this is the example for within the Salish Sea. So you can pull this kind of data out. Kate's better at leading you through how to kind of pull that out. I would say reach out to her if you have any questions, but there is a website and I'll share it at the end here. But I just wanted to show you some quick examples of the things you can do with the online data tools. I know Helen was involved a lot in the compiling of the data for the Salish Sea, so she could probably speak to some of this as well if there's questions. But you can also combine their substrate types and your habitat types to look at biotopes. So this is for Pacific Northwest. So you've got Washington, Oregon, and California. I'm spinning the mouse around there. The sub-regions of the Pacific Northwest ecoregion. And you can compare the substrate types here on the left with the biotic habitat types on the right. And so you kind of get at overlap within different habitat and substrate and biotic factors that might drive species assemblages, especially if you're looking in an area that's specific to your focus research or where you're trying to answer specific questions or find data on. So this is a repository of existing data. It's a compilation, and we're always trying to build on it. So there's obviously a lot of data gaps and limitations within the zones. We found that the intertidal zone mapping was definitely not consistent along the West Coast. Different regions, different agencies offered information, and that had to be kind of merged and married into the single online data tools. For habitats, we don't complete all of the CMEX components. CMEX has other classifications such as geoform and water column components that are not included here. For substrate data, we find that habitat classification levels vary significantly by their source and regionally. So similar to the zones, the source that was providing substrate data had different classifications of what is the size of cobble and pebble and boulder and things like that. So there was a lot to tease apart to get this into one online database. And the last example I give here is the biotic habitat. There are definitely some data gaps. There's primarily, there's a lot of data for habitat within the core zone, but it's very limited for the seaward zone. But again, that may be driven by the factors of depths and where is your photic zone with biotic habitats components. But for instance, macroalgae, there are a lot of data gaps associated with the understory, algal turf beds, coralline filamentous algae, and things like that. And the seagrass extent data were not as complete for the outer coast as they were for within Salish Sea. So well done, Helen, on that, of course. I think you were involved in putting a lot of that in. So Kate's constantly working on this. You can definitely reach out to her. She's always looking to improve the map application tools, improve the quality of the maps, include more data. We do hold in-person data tools training courses. The next one in February is at the Padilla Bay National Estuarine Research Reserve. I believe it's already full and there's a waiting list. But I think they're looking to hold at least one, if not two, a year. And they're usually held associated with the National Estuarine Research Reserve. So you can reach out to Kate or Joan Drinkman or myself to get on the waiting list and find out more information on those. But Kate asked questions about what do people want to see next? Do we want to expand our classifications for more of the CMEX components? Do we want to link spatial data with habitat and fish and things like that? So there's a lot of information that can still be improved on it. Okay, that was a lot. I hope I didn't go over too much. I can put these links and emails in the chat for you to copy and paste and grab if you'd like. John Drinkman is also one of the coordinators with PMAP that works a lot on the national level. I'm a coordinator working more with the steering committee and subcommittees, Science and Data Committee, and Kate is with Pacific States Marine Fisheries Commission as the data wizard. So if you just want to Google PMAP, you could go to PMAP's website. You could look up assessments and reports and download the report. It's 48 megabytes. And then there's also the link for the data tools and then some other data tools related to estuary. Okay, that was a lot. Happy to take some questions as we have time though.
I was gonna say, I think we have time for probably one question and then I'll direct you to the chat. So I don't see anything in the chat yet. So feel free to raise your hand if you have a question for Jamie. I think you've stunned them with the quantity. Oh, Helen.
Yeah, I said to Helen I would talk for like 5-10 minutes and then she gave me a half an hour. So all right, talk about everything then. Go ahead, Helen.
Helen, you're muted.
Oh,
there you go. I'd like to mention that Jamie's talk links really nicely to Dana's talk, which happened at the very beginning of this meeting, because Dana identified through the Pew study that we really need synthesized data to be available to managers. And so I'm looking forward to seeing how managers are using these data. And I think some of it can be pretty confusing for people who aren't intimately aware of all the considerations that go into those data viewers. So Kate does a great job, but it's just hard. So anyone who's interested in thinking more about this, and it may be through the context of Pew, those Pew studies, and I think Dave Lowry is here too. But that is a high priority for me personally, is how do we make these, how do we continue to get these online viewers of synthesized data out to researchers and managers? And I think this PMAP viewer that Kate's done is a great starting point.
yeah, sorry I missed Dana's talk. I had an overlapping meeting, so I came in late. I hope I kind of was able to provide a linkage there, but yeah, thank you.
Okay, thank you, Jamie. I think we are now going to move to our last presentation. So let me attempt to screen share. How does that look for folks? Okay, so Zach, just let me know when you want me to advance.
Elizabeth, those slides look familiar. Thank you. Let's give this one more try. Well, thank you, Elizabeth and Helen, for hosting and having us here today. I appreciate the opportunity to talk to you all. My name is Zachary Randell. I'm a research scientist at the Seattle Aquarium, and I'd also like to give a shout out to Megan Williams, a research technician. The two of us are now working on these set of research projects together. So the purpose of our work has been to harness the increasing accessibility of ROV technology, how it's become less expensive and more customizable, and to start to develop methods of using ROVs to conduct benthic surveys in relatively shallow waters, including in kelp forests. We're doing that, and we're also harnessing the increasing accessibility of AI algorithms to conduct image analyses. So the hope is to develop these analyses to process imagery at scale. So today I'm going to briefly touch on the ROV and talk about some of the work we're doing with the Port of Seattle and Elliott Bay, and then go over what we've learned about the ROV and these surveys and some of the metrics of the ROV surveys. And then I'll dig into a bit into more details in terms of the imagery that we're collecting, and touch on the AI analyses as well. Next slide, please. So this is the ROV itself. It's a Blue Robotics ROV. We have lights facing downwards, we have lights facing forwards, and we have cameras facing downwards and forwards as well. For data collection, we're using GoPro 10, so we're just strapping GoPros on. They collect high resolution 4K imagery, they're image stabilized, and they work well for us. In terms of sensors, we have a ping sonar altimeter, which provides a real-time feed of the ROV's altitude above the seafloor, which enables us to keep our imagery at a consistent scale. And also in the top left photo, you can see a top cylinder that's an acoustic transmitter. We're using Waterlink's acoustic system. And I see a question, are you running red filters on the GoPros? We are not, because we are providing light ourselves, primarily with the benthic facing lights. So we don't need filters, though it is something we could explore with the forward facing cameras, because the forward facing lights mostly make it look like a snowstorm in our waters, unfortunately. But the water, the ROV that is very small. It's really maneuverable. We can deploy it right off a small vessel. We just pick it up and plop it in the water. We pick it up to retrieve it, etc. Next slide, please. So one question that is justly raised is, are you going to be able to run this in kelp? What about the tether? Is it not going to get entangled? And we have been able to develop methods of running it into a kelp forest and then bringing it back out. We go straight into a forest. We don't try to do anything fancy, just straight in 50 or 100 meters. And then to come back out, we turn the ROV around, pan the built-in camera up, locate the tether, and then we stick very close to that tether. So we basically follow our trail back out of the bed. The ROV is small enough that it's able to maneuver to the left of a bed or to the right of a bed. And Megan just shared a link that you all can check out. It's a short video of us having the tether caught in the middle of a bundle of bull kelp stipes, and then by applying tension at one end of the tether and then maneuvering with the ROV, we're actually able to peel open a group of bull kelp stipes and then drive through the opening. So the key thing is this ROV is small and very, very maneuverable and responsive. I have no doubt that perhaps thicker kelp beds, I've seen some very thick kelp beds in the San Juans, for example. I don't know how the ROV will fare in those. We haven't tried it yet, but the kelp in Elliott Bay and some parts of the outer coast, it has worked well. Next slide, please. So in collaboration with Kathleen Hurley and the Port of Seattle, we have initiated a monitoring program throughout Elliott Bay. You can see on the right, 8 sites throughout Elliott Bay we've established. At each of these sites, we have 4 ROV transects each. And we just recently started sampling at the end of summer 2022. I'll talk a bit about some of the preliminary results for that. And we're also in the process right now of kicking off a winter season of surveys. So the idea in the summertime is we capture the benthic environment when kelp is at a maximum, and then we come back in the winter when the annual vegetation has been removed or is absent to try to get a better handle on the underlying substrate type, to better understand substrate availability, and in general, what species are present that associate with persistent vole kelp throughout Elliott Bay. Next slide, please. So probably the most buggy part of this whole thing thus far has been the acoustic tracking of the ROV. we're using a system by a company in Norway called Waterlinked, and they have an acoustic GPS tracking system. Sometimes it works really well and we get nice clean tracks like you see in the upper left. Sometimes, like you see on the left to the bottom, that's not what the ROV did, but we get funky readings. You can also see an example of funky readings on that second display on the photo to the right, where the ROV is kind of all squiggly with the red lines. That's not where we drove the ROV. So that's been buggy, and we've been working on it. If anyone out there uses acoustic GPS systems and absolutely loves what they're doing, let me know, because we're still working the bugs out of ours. That all being said, I still really wanted to map our transects. So over the course of last year, I developed some code where we take a starting GPS coordinate that was recorded properly, the compass heading of the ROV, which was recorded independent of the GPS and is recorded every second, and then a sample distribution of step lengths for how far the ROV travels as compiled by transects that were recorded properly. And we combine all that information, and then we can basically simulate or reconstruct our transects. So next slide, please. So these are examples of those simulated transects. This is in the East Waterway, just offshore of the Coast Guard station. So you can see the 4 transects, the 4 shades of purple are where we drove the ROV. And then you can see there's a bit of wiggle. There's differences in the width of the transects, and that's the actual area that we surveyed. And I'll come back to that briefly in a moment. Next slide, please. And this, just to give you another idea of what some of these transects look like, these are our 3 sites offshore of Centennial Park. So you can see we're driving underneath the grain elevator. Yeah, and again, note the little wiggle in the area covered. Next slide, please. So because we know the ROV's altitude above the seafloor, and we also know the angle of the camera lens, we're able to make some pretty simple calculations for the relationship between the two. And that gives us output of what's the width and height of the frame of reference that the camera films given the altitude of the seafloor. So we can then make the total area calculation, and then it's very easy to extend that to the entirety of a transect. So next slide, please. So what you see here is on the x-axis, that's transect time. So that's a continuous transect. And on the y-axis, that's seafloor area surveyed. So you see the wiggle on the y-axis. That's the various areas surveyed as the altitude of the ROV varies. And then that spike is the ROV actually driving over an underwater pipe. So the area drops because the pipe is suddenly below the field of view, and the ROV's altitude is just above it, so it thinks it's close to the seafloor. And then we maneuver up and over the pipe and are past it, and all of a sudden the seafloor is much further away because we clear the pipe. So that's what that spike is. Why I'm telling you all this, because we have these area calculations, we can then calculate other survey parameters to try to better understand these methods that we're developing. Next slide, please. And here they are. So these are frequency histograms for the 32 transects that we conducted as part of the surveys summer 2022 with the port. So the blue are the frequency histograms. The red line are the mean of these various parameters. So for those surveys, on average, our transects were a bit over 60 meters long. They took us about 9 minutes to complete. The ROV cruised on average at a speed of 0.12 meters per second. And in doing so, we covered on average 200 meters squared. And we had this metric of rate, which is just the total area surveyed divided by time. And that gives us a survey effort of 0.4. So what I'm hoping to do with these metrics is use these to compare and contrast with other survey platforms. And that's, next slide, please. That's actually what we're hoping to do next. So we are in the process of planning to do a diver ROV methods comparison later on in the winter and early spring, and then we want to repeat it in the summer. We're in the process of training some of the research divers at the aquarium on reef check protocols, and we want to go down, lay down meter tapes, have the divers get out of the water, send the ROV down, image everything, and then put the divers in afterwards We want to put the divers in afterwards because they're going to lift up blades, et cetera, and kind of not disturb, but silt up perhaps the substrate. So we want to get imagery before that. So yeah, I'd like to put it out to you all. If anyone wants to get in on this, I'd be very interested to do this in other locations and with other survey platforms. Let's see, yeah, so we were going to do this laying out the standard 30-meter diver transects, and we're going to do it just offshore of Centennial Park. Next slide, please. All right, so this is just a shout out to, we have figured out that we can run the ROV in Kelp, at least the Kelp in Elliott Bay, and we can indeed push against current. I don't think I mentioned that. By far, the #1 limiting factor, and this may or may not surprise you, governing image quality is water visibility. So we have figured out there are plenty of times of year or instances where it's just not worth putting the ROV in the water if you want to get good quality imagery of the benthos. July and August in the peak of the blooms may or may not be worth doing surveys. What we found was the latter half of August, September, and October, the water cleared up substantially, or there were instances where the water cleared up, and that's when we ended up doing the bulk of our surveys at the end of the summer. So we've learned a lot. We've been developing the lighting on the camera essentially from scratch, and we're now at the point where, next slide, please, Elizabeth, we can reliably capture decent quality imagery, dependent, of course, on water conditions. But we got the lighting and the cameras figured out, and if it's relatively clear down there, we can indeed pull out some good quality imagery. Next slide, please. I think I'm switching gears, yes, indeed, to data collection. So we have the forward-facing camera. This allows us to pull out metrics of abundance for fish and bull kelp stipes. And then next slide, please. We have the downward facing camera. This is more analogous to the standard diver swath and UPC, uniform point contact surveys. And let's see, I think Megan is going to share a link that you all can check out. That is right offshore of Magnolia. That's from the downward facing camera. And that gives you a sneak peek at what that looks like. I think the preview you'll see in Google Drive is like 1080p or something, but if you download it, it's the full 4K file. Next slide, please. I'll briefly just touch on, for those of you interested in the details, with the GoPro camera, the white balance setting, we were using auto for a while, which at times, or most of the time, produces really nice imagery. But unfortunately, we found it's prone to exhibit a green flushing. It kind of flushes to the green end of the RGB spectrum. That's what you see on the top left there. We can color correct for this. That's B in this slide, but it's very time consuming. So we've now switched the white balance to what GoPro calls the native setting. It's a more muted color. That's C, which does require correction in post-processing, but it's only you apply a single correction to the entire video as opposed to having to correct individual instances. Let's see, anything else I want to say about that? Yeah, the native setting does really, really well with varying light levels. It doesn't exhibit wide fluctuations at all. So we're able to pull out more consistent imagery. Next slide, please. Okay, and now for the analyses themselves. So I think I've talked about these in some of the past presentations. We're using two programs. They're both open source. Viami is used for object detection. So that's what you see with A and B there. So from the forward-facing view, we put boxes around the bull kelp stipes or bundles of stipes to train the algorithm. And then from the downward-facing view, as an example of that, you see sea stars and rockfish. And then we are also using CoralNet, an open source web interface program for percent coverage. I'm really excited about percent coverage in CoralNet. I used to do PSCO's UPC surveys back in the day where the divers lay out the 30-meter tape, and every meter you go through and collect data. So in one still image, you can put down 100 random points, or 50 or 200, whatever you want to do. We're using 200. And then you annotate, all right, is that fleshy red algae? Is that soft sediment? Is that brown algae, understory, et cetera? And then you train the algorithm to eventually predict those. Why I'm really excited about this is we have 100 points per still image. If you extract a still image across an entire 100-meter transect. So that's 100 still images. That's 10,000 data points. So this could drastically increase the amount of information we're pulling out about the benthos. We really need to understand these methods. Obviously, we're not looking under blades. We are not going to be able to do what a diver with hands and eyeballs can do. And that's why we want to better understand the methods by contrasting it with other platforms. But I'm excited to help develop this, and hopefully it will be useful. I think it'll be especially useful to better understand understory algae in particular. I think that's the bulk of it. Next slide, please. Yes, so everything we're doing, we're trying to make it as open source and accessible to folks as possible. So there's 2 different GitHub repositories. One is a more general information. There's a bunch of ROV videos we've posted. There's photos, there's interviews and documents. And then the other one to the right is more in the weeds. So we post any R code that we use is posted there, the coral net classifications. So the species we're pulling out or the taxa, that's posted as well as some information about our mapping. And yeah, I think that's it. I think Megan was going to share, yep, the link, there it is. I know it's also shared on the agenda, and the link to the second repo is right there on the home screen of the first. So you can find them both easily if you'd like. And then I think the last slide, next slide, please. Or close to last slide, just a quick shout out. The ROV is great for education and outreach. I don't really have time to detail what we've done thus far, but I can tell you that the ROV is small and portable. We can toss it in a pool, we can toss it in a marina. We've taken it out at sea with Macaw interns. It's been really cool to get students hands-on with the ROV. You control this thing with a Xbox game controller, and I can tell you some kids have picked it up that clearly have video game experience, and they are driving that thing around no problem. So it's really cool to be able to engage students and youth with this, and I'm excited to develop that further. So with that, next slide, please. Shout out to the Port of Seattle, North Pacific Coast, MRC, and Seattle Foundation and Trust for some funding, and thanks to many of you here for your support as we've gotten started this past year. It's been an absolute pleasure, and I'm excited to keep pushing this work forward. So with that, thank you very much.
Okay, we have about 3 minutes before we're done with the meeting. There's a lot going on in the chat, Zachary, so I'm going to keep that open once we're done so that you can look at that, and I'll also put it in the meeting summary. Any, maybe time for one question if you want to raise your hand. Brenda.
Hi, sorry, I'm trying to remember where all the buttons are on the different platforms. Zach, thank you. That was great to see your presentation. And I popped my question in the chat, but I'm really curious about applicability and specifically how your work can help inform the ongoing efforts that are currently underway to help the state identify and cite and prioritize areas for kelp and eelgrass conservation and restoration. Specifically, I wouldn't say specifically interested, but really interested in how your benthic mapping could help inform restoration, identifying best restoration sites.
Yeah, thank you for that. It's a great question. I think to a certain degree, I would almost bump it to the relevant state officials that are doing that work to identify the greatest needs that we could possibly fill with the ROV. What I will say off the top of my head, that specific substrate types, like fine scale details about substrate type, that's something we can pull out from the ROV video easily. And that's something that we're training the coral net algorithms, the percent cover, to detect. So in addition to species, I think I mentioned this, but silt, shell debris, hard substrate, anthropogenic structure, we're starting to train those fine scale substrate types into the algorithm. So we will have metrics of those in addition to the other taxa as well. Yeah, so, and I'd open it up if anyone else wants to jump in with how those types of data and what else we're collecting could inform potential restoration sites.
If I could jump in really quickly, this is Kathleen Hurley from the Port of Seattle, and just as a context of how this work is helping us, in terms of mapping and to your question, Brenda, it's really helping us understand what nearshore aquatic resources are within our properties, and then ultimately understanding what substrate and which environments kelp are growing in this particular environment in Elliott Bay will help us inform how and when we could incorporate kelp enhancement in future nearshore restoration projects. So that's one application that we at the port are find that this is useful for, hopefully.
Okay, so we are at time. Thank you, Zachary. That was wonderful. I am going to keep the chat open for a little bit so that you can, anyone who wants to add last thoughts, or if you want to read through those links, please do so. Otherwise, this will be posted on the Nearshore YouTube channel soon if anyone wants to re-watch the videos. And I'm going to hand it to Helen for any last thoughts before we adjourn.
Thank you so much, everyone, especially to all the speakers. And I'd just like to say that we have another amazing list of speakers that will be speaking on May 24th. I'll send out a draft agenda in a few weeks, but please let us know if there are things we're missing. So anything you want to hear about or anything that we don't know about, someone we should have speak, let us know, and we'll see you on May 24th.
Right. Thanks, everyone.